So here we're going to talk about marine microbes, and that's going to be viruses and bacteria in the marine environment. This is going to be, first of all, a comparison between prokaryotes and eukaryotes, which you probably remember from biology, but if not, this is definitely a good thing to review and make sure you have those differences pretty well down. So viruses actually play a massive role in the marine ecosystems, just like they do in our own lives. You catch the flu bug, you catch a stomach virus. These are obvious viruses that we're used to hearing about, but you're probably not used to hearing about the ones in the marine environment. Some funny things about viruses, they're technically not considered living things, or maybe they are. There's actually some debate about this. The reason being is they have no metabolism. So they don't metabolize, and that's something that's usually considered to be a major characteristic or a requirement of being a living thing. And basically, it can only reproduce inside a host cell. So it can't reproduce on its own. It needs to invade a host cell, whether it's a bacterial cell or an animal cell or a plant cell, in order to reproduce itself. So there's basically two theories on what viruses are. They're either very, very, very simple prokaryotic parasites, or they are just renegade genes, because viruses are basically just a package of genes with protein coat around them. So the interesting thing about viruses is that all viruses are pathogens. Pathogen means a disease-causing agent. Not all bacteria are pathogens. You've heard of good bacteria, bad bacteria, and we'll talk about that in a minute, but viruses are all bad in the sense that they all cause disease. Now, viruses attack bacterial cells, and these are called bacteriophages, is a virus that attacks a bacterial cell. So there's a cycle that they go through. So we're going to look at the virus's life cycle here. And that's known as a lysogenic cycle. And that's basically when they reproduce their DNA inside of a host. Then there's another cycle called the lytic cycle, which is where they actually assemble themselves inside the host and then break free from that. So here's an example. This is a Vibrio cholerae. So you've heard of cholera probably. That's the virus, or I'm sorry, that's the bacteria that's in the human body already. So you might not know this, but a lot of the bacteria that you hear about causing diseases, you already have those in your body all the time. And having them does not cause disease necessarily. It's when they get too numerous or when a virus attacks them. And that's what happens here with cholera. So you have Vibrio cholerae in your body, and it's non-pathogenic until the virus changes its DNA into something else. So the virus, the bacteriophage, attacks it. So here's the lytic cycle. So we'll start here. We'll start at this point where a virus latches on. This little green thing is the virus, and this black ring here represents the bacterial cell. So here's your bacterial cell, here's your virus. The virus latches on, and the little red string, that's the virus's DNA. So the virus sticks its DNA inside of the bacteria, and this blue loop here, that's the bacteria's DNA. So now the bacterial cell has its own DNA, and it has the virus's DNA. And during the lysogenic cycle, the virus's DNA actually merges itself with the bacteria's DNA. So now the DNA is basically one type of DNA. So it's changing the bacteria's DNA. Then the bacteria goes about its normal way of reproducing. And with bacteria, that is by division. They basically, it's asexual. They divide into two. And we'll talk more about that in a minute as well. So now there's going to be two that have the virus's DNA embedded within the bacteria's DNA. And then the cycle continues. So eventually what happens is that DNA that's, you see the little red part here, is going to code for the making of more virus. So DNA codes for making proteins, which you probably remember from biology. So DNA codes for, here's the virus's DNA. Now it's separate, okay? So it's going to code for making more virus. So now basically the virus has hijacked the bacterial cell and is using it to reproduce and make more virus. So then you have the virus proteins that are assembled within the bacterial cell, and then they burst forth during the lytic cycle, and it kills the bacterial cell and it releases a lot more virus. So that's basically how a virus works. It hijacks a host cell. So in the marine environment, we see a lot of viruses. A small sample of seawater contains as much as 100 to 10,000 different types, different species of marine viruses. So that's amazing. Most of the time, you think about swallowing seawater now, it might feel a little bit different, but basically your body can fight off these viruses 99.9% of the time. It's when they're too numerous or when they're too strong. Maybe they have a resistance to your body's own immune system that they can be harmful. So the marine environment, of course, other animals and plants and microbes in the marine environment are affected by them. Algae in particular and other single-celled eukaryotes can be affected by viruses. So we'll see a little bit more about viruses later, but now we'll look at bacteria too. So bacteria are within the domain eubacteria. They fill pretty much every niche you can imagine. They can be primary producers if they're photosynthetic or chemosynthetic. They can be decomposers, they can be food, they can be involved in symbiosis, they can be pathogens, and they can be an important part of certain biochemical cycles, which, if you think about on land, that happens a lot with decomposition in the ground, which kind of links into decomposers, or nitrogen fixation, which is the way that a lot of plants like legumes, which include peanuts and lentils, they depend on nitrogen fixation by bacteria. So here's a little review of what their cells look like. So this is quite a bit different than a eukaryotic cell. So you have no nucleus. You just have DNA that's kind of jumbled up into what's called a nucleoid region. So there's no nucleus. They have little tiny ribosomes, but they don't have all those other big organelles that you see in eukaryotic cells. They don't have mitochondria. They don't have a Golgi apparatus or an endoplasmic reticulum. They have none of that. They do have a cell wall. So that's something that plant cells have, but animal cells don't. And bacterial cells, obviously, they've got their cell wall here. And oftentimes they're also surrounded by a capsule, so it's just another layer of protection. They move around by these pili and maybe a flagella, or one or the other potentially. And bacteria reproduce by way of binary fission. So we saw a little bit of this when we were looking at the bacteriophage, the virus attacking the cell. But binary fission is basically when it asexually splits from one cell into two. Okay, it's pretty easy, pretty straightforward. The DNA undergoes replication, makes a copy of itself. So now you have two little cells starting to split off here, each of them with identical DNA. And they split, and then they're suddenly two completely separate cells with their own DNA. So it's basically cloning. It's basically the parent cell divides itself into two, and then so on and so forth. And this can happen insanely fast. They can do this in a matter of seconds or minutes. So that means that bacteria have the ability to evolve very quickly. So something else that's important about this is even though the daughter cells technically should be identical, they're not always going to be. And the reason for that is mutation. So mutations are going to crop up during this part of the phase here when the DNA makes a copy of itself. So there could be subtle differences between these two. And that's how evolution occurs, because sometimes, every now and then, that mutation might confer an advantage to one individual cell over another, and that cell might be selected for based on the environment. And because they reproduce so often and so fast, it can happen very quickly. So bacteria can evolve extremely rapidly. So the way bacteria eat, the way they get nutrients, is pretty straightforward. They can absorb nutrients across their membrane, and what they do is they can secrete an enzyme that breaks down the nutrients outside of their body and then absorb it across their membrane. So it's kind of like they digest things outside of them, and then the nutrients are able to move across their cell membrane. So that's called osmotrophy. So think of osmosis moving across the cell membrane, and that's what's happening there. Another category of bacteria that's really fascinating are cyanobacteria. So these are photosynthetic marine bacteria, and they're known as blue-green algae. What's amazing about cyanobacteria is they are some of the oldest organisms on the planet. They've been around from the very beginning. They think that the earliest prokaryotes were a type of blue-green algae that could do photosynthesis, and that's what these guys can do. And that's because they have chlorophyll pigments in them. That should say chlorophyll A and chlorophyll B, but that's the little pieces are not working there. So chlorophyll A and chlorophyll B. Cyanobacteria can be found in huge mats, these huge microfilaments that you see here. So you can actually pick it up, and it's like this gooey, spongy feeling to it. They can cover the bottom of lakes. They can also fossilize colonies of them. And this is amazing. This is something called stromatolites. They're found mostly in Australia. And these stromatolites are basically fossilized cyanobacteria colonies. So basically down here at the bottom would be the oldest fossils of cyanobacteria. And get this, some of them are 3.4 billion years old. 3.4 billion years. They are almost as old as the Earth itself. So you can look at these things. If you go down to Australia, you can actually snorkel around with these things. They're just large, hard. They look kind of like mushrooms. The bacteria that's on the top layer is living, and underneath is where it's fossilized. So imagine living on top of your ancestors, going back as far as life can even go. That's what these bacteria are doing. So they just, as they die, they just fossilize and continue to build up on these mounds. So it's pretty remarkable, these mats. The way they fossilize is the way anything fossilizes. They trap sediment, and that's the hard part that becomes hard and calcareous. So pretty amazing. Bacteria can be categorized kind of by how they tolerate different abiotic factors in the environment. Oxygen tolerance, for one thing. You have what are called obligate anaerobes. That means they cannot have oxygen, like oxygen is toxic to them. Facultative anaerobes means they can do respiration when oxygen is low, and they can photosynthesize anaerobically when light is present. So these guys can take or leave oxygen, basically, is the way of putting that. Bacteria organisms that can do photosynthesis contain what are called bacterial chlorophylls. So this is different than a plant cell that has a chloroplast. Chloroplasts do photosynthesis. So these guys have bacterial chlorophylls, which are little pigments that aid in that process. Chemosynthetic bacteria, of course, are kind of a different category, but they can make their own energy as well. So think about chemosynthesis. You've talked about this a little bit before, so I won't spend too much time on there. But that's where they use dissolved chemicals in the vent water that comes out of hydrothermal vents as their primary source of energy to get that ion to make energy. Less efficient than photosynthesis, but it gets the job done, definitely. And you have a lot of life that thrives around them down there. So not all bacteria are producers. Some are heterotrophs. So decomposers will break down organic compounds into inorganic molecules for reuse. There are free-floating bacteria that aid with certain sediment processes. Lithification is basically where the pH of the substrate or the mud or sand is changed, and that causes minerals to leach out. Consolidation is a process where particles stick to each other because of electrical charges, and bacteria can be involved in that as well. And then sedimentation is when particles will aggregate and then settle into sediment. So, for example, if you have a cup of coffee and you've dissolved sugar in your coffee, you think you've dissolved it, but then after drinking it, you see there's some sugar that's on the bottom. Maybe it did dissolve, and then as your coffee cooled off, as it got cooler, the sugar basically precipitated out. And so that would be like sedimentation. Other processes, the nitrogen cycle, which we'll be getting into more detail later, but the nitrogen fixation cycle is something they're a part of. Nitrification is another thing. Primary producers need ammonium to be converted to different forms of nitrate, so that's another part of the cycle. Then there's symbiosis, and this is something really remarkable. There's something called an endosymbiotic theory, which is basically the idea that modern-day eukaryotic cells are basically a symbiosis between an ancient prokaryotic cell that got engulfed by the larger cell, and now today they work together. So this is amazing. Mitochondria, which are in every cell of your body, they're considered to be an organelle, are actually were once their own prokaryotic cell. And proof of this is that they have their own separate DNA, which is pretty amazing. Same thing for chloroplasts inside of plant cells. So chloroplasts and mitochondria might have once been their own independent prokaryotic cell that just got sort of enslaved, if you will, by a larger cell. So there's a couple of hypotheses here that there's a hydrogen hypothesis that these early prokaryotes like mitochondria were hydrogen producing, and the larger cells needed that hydrogen, so they engulfed them. They think that might have, for example, happened in the gut of these tube worms with the bacteria that can do chemosynthesis. There's also bioluminescent bacteria, so these guys will emit light, which is pretty cool. We'll see more of them as we go as well. And then finally, archaea are the last marine microbe. They're smaller than bacteria, they are prokaryotes, and they're basically very similar to bacteria, except for they tend to be extremophiles, which means you'll find them in very extreme temperatures or pHs or salinities. Some can even metabolize methane, and that's methanogens. So that's kind of cool too. So anyway, that kind of summarizes our prokaryotes, and we'll be looking at plants next. in the next slide, and that'll be under 19.2.